Okay, that's right. So I'm ahead of myself. That's good. All right, tell me, are you guys seeing this lecture, chapter 13?
Yes.
All right.
The last notes I have are on slide number 19.
Taking notes where I'm at. All right, before I get started, let me just start with a prayer, please. And then I'll ask them if you guys have any questions, address things that you might have, and then I'll just complete this chapter and we're done. Heavenly Father, God, thank you so much for what you do for us. Thank you again for this week. Thank you again for our students. Watch over them and their families by extension, oh God. Keep them safe and well. I know we're almost halfway through the semester. It's incredible. I think we're approaching week 6 or 7. And with exams coming up, midterms, and so on, I know our students, man, they have a ton going on. Keep them focused, oh God. Help remind them of their purpose in life, things that you've shown to them, and help them to achieve that goal. To always be mindful of their studies in relationships, friendships, parents, grandparents. What we see things tenuous, oh God, especially during this time. Watch over them all and keep them safe and well. In Jesus' name I pray, amen. All right, guys, let me set the day up for you. I'm going to complete this chapter and that's it. We start chapter 14 on Wednesday. Just a couple things. The exam is graded. This is exam 2. The average was 78.5, somewhere about 78.5. So we're doing pretty good with scores. Keep it up. I want you guys to start thinking about the next exam. I know you don't want to hear that, right? But excuse me, you have to move on. So exam 3, pay attention. It covers 2 different chapters again. These are 2 chapters. So it's chapter 13, which we complete today. And then chapter 14. So chapter 14, we're going to start on Wednesday. So my objective today is going to be a very brief lecture. I do want to finish up this chapter. And then I haven't scheduled an exam yet for chapter 4 exam. Well, I haven't scheduled exam 3 yet. Once we complete chapter 14, again, I'll give you a week's notice, all right? Just like we've done before for all the exams. So today I do want to complete chapter 13. Before I get started with the remaining part of this chapter, I want to open up for any questions you might have.
Professor?
No problem. I'm sure you do.
Excuse me, Professor?
Yeah.
Do you mind posting the hour-and-a-half lecture instead of the hour lecture on Blackboard?
Okay, so I'll check my WebEx again and see if it's there. Hopefully, so last time, well, the lecture I posted was from this section, actually. Remember, we didn't have a full lecture because we did a little review for the exam, right?
Okay.
Yeah. So I'll record this one too, and I'll post the end of this one today. It takes a while for WebEx to actually create a file that I can download, so you probably won't see this today, all right? But it should be up in about a day or 2. Are there other questions I can address?
I have a question for the exam.
Yeah.
So is the exam going to be kind of like the last one where it's like name, symptoms, origin, and transmission and all that?
Pretty much. Are you talking about exam 3?
Yes.
Yeah, so it'd be the same type of format.
Okay.
Multiple choice, important things, names of organisms, you're right. Disease-specific symptoms. And if I tell you a treatment, I'd like you to know that. If there are vaccines to prevent certain diseases, I'd like you to know that too. But I don't ask you to memorize antibiotics. Many different reasons why. It changes depending on what hospital you work for. They tend to have their choice of antibiotics. That's one reason. The second thing, sometimes it's very difficult to predict what antibiotic would work because bacteria are promiscuous and their resistance and sensitivities change. All right?
All right, thank you.
Good. Other questions I can address?
I just wanted to make a note. The last slide that we left off on that I have notes on is 19.
19?
Mm-hmm.
All right. So this is this disease here?
Yeah, that's the last one.
Right. So this is just this is information just related to that. Landa's disease I don't talk much about because we haven't seen this disease since 1945. It's something you have to don't be concerned about. In the United States, we haven't seen this. This is primarily the disease of horses, actually. But occasionally humans do get infected. We've taken care of this problem pretty well. So we're done with this chapter for soil-borne diseases. Other questions I might address before I move on? All right, guys. So here's what I want to do. I want to complete this chapter, and then we're done for the day. So just to kind of remind you what we've been doing, this chapter deals with soil-borne and arthropod-borne diseases. So we looked at the major ones. We talked about tetanus, for example. We looked at gas gangrene. We looked at melioidosis. We looked at leptospirosis, right? What I want to move on next to are diseases we acquire through a vector. That vector, it's tend to be arthropods. So what are arthropods? These are arthro means jointed. So these are jointed creatures. Think about things like ticks, lice, and fleas. Yes, they can, even mites, they can transmit disease to humans. The bacteria themselves do not cause infections in these animals, like ticks, mites, and fleas. But the fleas feed, or these animals, they feed on other animals that contain the pathogen. And then they suck on the blood, or they feed on their tissues, and then they bite you. So you can actually transfer the disease from animals to humans. Those are zoonotic diseases, but it requires a vector. And the vector in this case, again, would be ticks, lice, fleas, or even mites, all right? Now, the major diseases are shown back here. So this is what I'm going to focus on today. Plague. You probably hear about the bubonic plague or the black plague caused by the same organism. That's Yersinia pestis. It's a gram-negative rod. I'm going to talk about some signs and symptoms for disease. It's a very nasty organism. In fact, if it's untreated, depending on the type, especially if it's a respiratory type, that's called the pneumonic plague, 100% of people would die. So it's a really vicious organism. The modes of transmission, you notice, through flea bites. So I'll introduce that flea to you. It's called Genophilus chiopis. That's the name of the flea. Don't worry about scientific names, but flea is fine, all right? Treatment, don't worry about, right? You're not significant here. So the next disease is tularemia, caused by Francisella tularensis. Other disease, I'm sure you heard about it, Lyme disease, caused by Borrelia burgdorferi. I know we mentioned that name in the laboratory, right? This is a spirochete. It looks just like Treponema pallidum, the one that causes syphilis. And then we get into the rickettsial diseases. So right over here, rickettsial diseases. Rickettsia are very important organisms. They're very kind of strange because they're very small. And again, literally, you can't see them with a microscope, the ones that we use in the laboratory. So we have to use electron microscopes to view them. They are obligate intracellular parasites, meaning they require living cells to grow. So I'm going to mention a few important species. The one that causes Rocky Mountain spotted fever, that's Rickettsia rickettsii. The one that causes epidemic typhus, that's called Rickettsia prowazekii. The one that causes typhus, which is Rickettsia typhi. And then one we see primarily in Asian countries, the eastern part of the hemisphere, the planet, that's Rickettsia, or it's now actually called Orencia tsutsugamushi. And then, of course, I'm going to talk about ehrlichiosis and anaplasmosis. In between, I'm going to talk about an organism called Borrelia hermsii. So we talked about Borrelia burgdorferi, that causes Lyme disease, but hermsii causes relapsing fever, all right? But notice this, they're all transmitted by arthropods, either a tick, lice, flea, and sometimes mites, all right? Now, the first disease I want to talk about is the bubonic plague. And again, many different names to this, bubonic plague, the black plague, it's all caused by the same organism. That organism is Yersinia pestis. Again, if you go back in history, of course, people didn't know this bacteria existed. They didn't even know microorganisms existed, as you well know. But based on the description of these diseases, scientists can backtrack and say, you know what? What actually affected people or populations during these periods, as I have listed here, was Yersinia pestis. In other words, the black plague or the bubonic plague. So if you go back to 542 AD in Africa, this lasted for 60 years. So people were describing this disease almost to the T, actually. Millions of people died. And again, don't memorize history. It's not significant here, but I do want you to understand how significant the pathogenesis of a very deadly organism. You go back to the mid-centuries, the black plague as it's called, defined because people who had this disease had black splashes on their skin, necrosis on the skin. But Check this out, 40 million people died in Europe. That was about a third of Europe's population estimated at that time. And again, you can go on all the way. Even in 1900, we had isolated in San Francisco, but that was that event originated from rodents transferred from the Far East. All right. Now, a term I want you to know is the sylvatic plague. Sylvatic refers to the wild. So this is plague that you find in animal populations. Now, you don't hear about the black plague much anymore. In fact, you don't hear about it at all, or the bubonic plague. Here's the reason why. We separated ourselves from wild animals. So check it out. Rodents, right here, you look at rats, mice, prairie dogs. They can actually harbor the organism that causes the plague. That organism, again, is Yersinia pestis. So we've separated ourselves pretty well from the wild. Think about that, right? Now, if you were to trap animals in the wild, rodents in particular, you can still find the organism. So don't ever think the organism has disappeared. That's not so at all. We're going to find out later on when we start virology, the only disease we've eliminated from the world's population or from the human population is smallpox, and that's a viral disease. So again, this organism, this bacteria, Yersinia pestis, still exists, but you find it in the wild. All right? So generally, when you see a plague case in humans, it's because they came in contact with fleas that bit an infected animal that causes zoonotic disease. All right. So again, if you take a look at each of these dots, this is the continental United States. Each dot represents one case. Now, again, if you go back to the mid-centuries, you find about millions of cases, actually. We haven't had a lot of cases for a very long time. So this is from 1970 to 2012. That's a long time. And very few cases compared to something like the mid-century, right? Again, the reason why is because we've separated ourselves pretty well from rodents. Now let's talk about the disease itself. The organism that causes the plague is Yersinia pestis. So somebody asked earlier, what do I want you guys to know? Well, names of organisms for sure. Yersinia pestis causes the plague. There are different forms of the plague. We'll talk about them in a second. But let me just say a few things about the organism. First of all, it's a gram-negative rod. Now, this is not a gram stain. You guys all know this gram-negative organisms. They stain red or pink or orange, right? This is not a gram stain. This is called a Giemsa stain. I have this to show you one thing in particular. Take a look at the organism. This is the bacteria. This is a blood smear for an individual who has the plague or who had a plague. So you see red blood cells in the background, you see white blood cells here, and then you see these strange looking organisms. They look like little safety pin. That's called bipolar staining. When you stain the bacteria with these stains, you see two little dark staining spots on the poles or on the very end. And that's called, again, looks like a little safety pin. It's called bipolar staining. So this organism, gram-negative rod, right? But it stains bipolar. Now, there are two major forms of the plague. The first is called the bubonic plague. I'll tell you the difference in a second. And the second is called the pneumonic plague. Well, the second one is kind of self-explanatory. Think about it. Pneumo refers to the lung. So again, this is a respiratory disease. Let me just say both are pretty lethal, but especially the pneumonic plague. If it goes undetected and untreated, check it out, 100% mortality. All right, so this is terrible. This is a very vicious pathogen. There's bleeding in the lung. There's cardiovascular collapse. People die. And of course, you have necrotic tissue. So at some point, the bacteria gets into the bloodstream, and then it's just circulated throughout systemic. All right? So the other form is the bubonic plague. Bubors refers to swollen lymph nodes. And I'll show you a second. I'll show you a picture in a bit. So again, from the bloodstream, the organism can get into tissues. It can float around in lymph, and then they can actually end up in the lymph node. So the organism is present in the lymph node. This is a very dangerous form also. Now less virulent, but still quite so, compared to the pneumonic plague. So about 100% for the lung disease, which you'd expect that's a very critical infection, about 60% for the bubonic plague. All right? Now, regardless to what, it doesn't matter if the bubonic plague or the pneumonic plague, at some point the bacteria can actually enter the bloodstream. And we already know this. When you have microorganisms floating around in the blood, basically it's dispersed all over the place, right? So now you have infection in the heart, the liver. Basically, you have systemic sterile sites, normally sterile sites that's infected with the organism. It's a very nasty organism to have. In that case, we refer to as septicemic. So septicemia simply means this. I have bacteria, which is Yersinia pestis, and there are toxins floating around in the bloodstream. Very, very dangerous, of course. All right, don't worry about treatments. We can use tetracycline or streptomycin to treat this, but don't memorize that fact. Now, let me show you some pictures back here. This is, first of all, this is the flea that transmits this. So think about it. That flea is called Xenophila chiopus. Here's what happens. This flea feeds on an infected mouse, or let's just say a prairie dog, a rodent. And just imagine that rodent has this organism, Yersinia pestis, in the bloodstream. So this flea picks up that organism from the rodent, and now it bites a human being. Of course, what you can see now is a very dangerous infection. So let's take a look at the infection back here. This one is called bubonic plague. Buboes are swollen lymph nodes. You can see this on the leg, and you can see this on the neck, the cervical area here. Now, remember I said fever is a very general thing, but sometimes we have some specific symptoms. If you see this thing, like rose spots for typhoid fever, right? That's a very specific correlation. If you see these types of symptoms, then you say, maybe this is the plague. Well, here's a specific thing I want you to know. First of all, for bubonic plague, you have a swollen lymph node, and then you see these dark patches. These are splotches, like purple or dark purple or black splotches on the skin. Now that's kind of interesting because it's almost uniform, all right? You don't see it too well back here, but you do see these dark purple splotches on the skin and bleeding that can also occur in this area. So not only are the lymph nodes swollen, but you see these pigments on the skin. All right. So again, keep in mind, doesn't matter if it's bubonic plague or pneumonic plague, the organism can end up in the bloodstream, and now you have septicemia. All that means septicemia, septic is pretty nasty. You have the organism and their toxins floating around in the bloodstream. Now, this is the last case I know of right here in the United States. This was in Oregon, maybe about 2 or 3 years, actually maybe about 5 years ago. Paul Gaylord. What you can see right over here, take a look at this. Remember the Black Plague? What does that mean? Well, the organism destroys tissues, and at some point you have these black spots on the skin. That's necrosis, actually. That's dead tissue, all right? Very toxic organism. And again, I have an expansive list here of the types of symptoms that you see. So again, pneumonic plague, fever, of course, but fever is just a very general thing. Headache, malaise, bubonic. Remember what bubonic means? That's swollen lymph nodes. Not with every disease you get swollen lymph nodes. That's not true at all. With viral diseases, there's something called EBV, Epstein-Barr virus, that causes infectious mononucleosis. So you can see swollen lymph node. But this is not a characteristic for many different diseases. So again, you see swollen lymph nodes, you see spots or those splashes, purple splashes on the lymph node area. You're dealing with the plague, all right? Of course, the pneumonic plague, think about this, the lungs are infected. Of course, you're going to see symptoms related to ventilation and respiration, all right? Okay, so again, very general symptoms like fever, diarrhea, chills, but keep in mind, you see necrosis of tissues, pretty bad, all right? Or you see buboes, swollen lymph node with spots or splashes on the skin, or splashes on the skin. That's typical for this disease. Good thing we don't see too many cases again, very, very few or none per year. Now, the next disease on the list is tularemia. Emia, by the way, refers to blood, all right? This bacteria was discovered right here in California, a place in a county called Tulare County. You probably know about that county. Anyway, so it's named after Tulare County. The organism that causes tularensis, the organism that causes tularemia is Francisella tularensis. Again, it's a gram-negative rod, and just like we saw with the plague one, Yersinia pestis, this organism stains bipolar. Take a look at it. It looks like a little safety pin. That's the characteristic staining that we see. You see that on a gram stain. Again, this is not a gram stain, it's a Giemsa stain. And so this is characteristic for the organism. So what do I want you to know about tularensis? It's caused by Francisella tularensis. Typical is transmitted by ticks. So here's what happens. Ticks can feed on animals again that have this organism circulating in the bloodstream. And then that tick bites you. All right, don't worry about the scientific name of the tick. That's not significant here, but tick is the vector. People have also actually acquired this disease by skinning. So imagine here's a hunter catching rabbits or some other animal, skinning, and just accidentally stab himself or herself. You just transported that organism from the infected animal to humans. You can even consume meat that contains the organism, and then you can get the disease also. All right? So what do you see in patients who actually have tularemia? Well, first of all, this is the tick. These are the ticks that bite. They carry the pathogen. If you notice that the side of pathogen also develops. So here's an example right there. This is a fresh one. Now, many times what happens that resolves on its own. In other words, people have a pretty cool defense mechanism. The bacteria doesn't proceed further, so there's no further infection. But in some cases, what happens, that organism can penetrate into the bloodstream. And of course, when you get to the bloodstream, as we mentioned before, that's a terrible game that we have. So the organism can actually get into lymph, in other words, tissue fluid. It can actually go to the lymph nodes. We have lymph node swelling. But notice this, you don't see those splotches, those purple splotches on the skin. So this is one distinguishing feature in comparison to the Yersinia pestis that causes the plague. All right, so again, generally speaking, you can see swollen glands for inhalation tularemia. Basically, the organism gets into the lung. Now you have a respiratory disease. So what do I want you to know? Francisella tularensis causes tularemia. It's transmitted via a vector. That vector is a tick. So basically, that tick bites an infected animal. It's a zoonotic disease, right? Bites that animal, gets the bacterium, and then transmits it to the human. All right, I'm going to spend a little more time on this one because it's a very significant disease. I'm pretty sure we've all heard about this disease. It's Lyme disease. Now, that's a strange name, right? Well, this Lyme disease comes from the term Lyme, Connecticut. So basically, first time we saw this disease was back east. This is back in 1975. Guess where? In Lyme, Connecticut. Hence the name Lyme disease. Now, the organism that causes Lyme disease, in fact, I had you guys memorize this for the lab practicum, like the spirochetes. Treponema pallidum causes syphilis. Borrelia burgdorferi causes Lyme disease. It's a spirochete. Here's a picture of that organism. It's a spirochete, all right? Again, first time we saw this was back in 1975 in Old Lyme, Connecticut. Mostly, it's the most, check it out, the most commonly reported arthropod. We're looking at ticks now, borne disease in the United States. So we see an increase in cases over time. Initially, we saw this back in the Northeast. If you notice, kind of spreads all the way then down south to the southeastern part of the country, but kind of strange. We don't see a whole lot of cases in the middle part of the country, probably because the vector is not there, all right? And of course, in California, we see practically everything on the West Coast. So the East Coast, West Coast, and then southeastern part of the United States. That's where we see most cases. But again, the concentration is back east. All right. This disease the bacteria is transmitted by a tick. It's called a deer tick. Scientific name is Ixodes scapularis, and the one that we see here in California primarily is Ixodes pacificum. Name. Think about the term Pacific Ocean, right? Pacificus. I don't care that you know the scientific name. I do want you to know what transmits this to humans from animals. It is the tick. All right. When we take a look at Lyme disease, we have to consider three different stages. All right, so we have stage 1, stage 2, and stage 3. And there's some critical telltale signs, especially for the first two phases or stages. You know what? If you know the patient's history and you see these things, you don't always see them, by the way, but you see these types of symptoms, I say, wow, I don't have to think about anything else. That person has Lyme disease. So what are those critical things, all right? So we're going to consider the three stages. First of all, think about this incubation period can be very short or very long. When I say very long, within maybe about a month or so. So here's what's going to happen. The early stage, this occurs in a matter of days. So check this out. The person's been walking in the forest, or maybe it's the wooded areas. That's where the ticks are, right? That's where the infected animals are, rodents in particular. So what happens, that tick bites a human being, and then that person comes back home in about 2 or 3 days, or maybe up to 4 weeks or so. This is what you might see. Now, not all patients, again, have this sign. That is called a target sign. Of course, it looks like the target sign, right? It's also called a bullseye rash. So think about it. That's exactly what it's showing, bullseye. Now, this is a pretty cool one. I don't think you can get as good as this one. You see that thing, you already know what that patient has. So when we look at stage 1, this is the early localized stage. It's characterized by something called erythema migrans. Migrans to move. Erythema means reddening. So this thing is expanding. All right. So this circular expanding rash that occurs can grow up to about 15 inches in a week. So this is erythema migrans. All right. Okay. So again, I want you to make that link. You see a bullseye rash or a target sign, you start thinking, given the patient history, the patient's been in the woods, you start thinking Lyme disease. Now, I want to caution you again, not everyone shows this symptom. In fact, the symptom doesn't always have to look like this perfect target sign. Take a look at this, might just look like a rash on the skin right here, maybe a fungal rash, tinea or ringworm. So it doesn't always appear to be that nice, perfect target sign. Okay. At some point, a person enters stage 2. And again, just depends on the patient, the immune status of that patient. But this can occur in weeks to months. And what happens, of course, this is erythema migrans, that rash is spreading, again, about 15 inches in a matter of weeks or so. But here's what's happening, just like we're going to find out with syphilis. If this goes untreated, right, again, the best time to treat this disease is in the primary stage. At stage 1, you have very few bacteria to begin with, and we have pretty effective drugs for it. But if that's missed, here's what's happening. That bacteria actually is penetrating into tissues. It's in the bloodstream, and from the bloodstream, it can go to different tissues, including the heart and the liver, and even the central nervous system. It can infect peripheral nerves, so this could be a huge problem. Again, the earlier you take care of this problem, the better is it for the patient. So again, the skin, the heart, nervous system, even synovial fluid. You can find the organism in synovial fluid, which is joint fluid, right? So this is the early disseminated stage. And then the third stage is called the late stage. This can occur to months to years, and this now becomes a chronic disease. And think about this. You have these spirochetes, again, they're moving about in the body. They can start penetrating and even replicating all pretty slowly. They can start dividing and increasing their numbers in the human being. So again, if you take a look at the stats, about 10% of individuals, this is 10 in 100, 1 in 10 person actually can develop chronic arthritis. So again, take a look at this right here. Right, this is the knees. You can see it's arthritic, it's swollen. Take a look at the joints back here. So there's a pain, there's swelling of the joints. All right. Okay, there's a vaccine for dogs. Dogs are very susceptible. And by the way, take a look at this right here. That's a target sign. This is in a dog. So there's a vaccine for dogs. It's still around. There used to be a vaccine for humans, but because there were so few cases, relatively speaking, they pulled that from the shelf because it wasn't selling a whole lot. But nonetheless, we do see an increase in the number of cases of Lyme disease, well close to 100,000 or so per year. Most of them, again, occur in the northeastern part of the country. All right? Now, to understand this disease, you have to understand the life cycle of the tick. So remember, the ticks are transmitting this from infected animals to humans. So let's take a look at this. This is a life cycle of the tick. You notice we have 5 different animals involved here. We have deer. The deer tick is called Ixodes. Here's the tick itself. We have, this is the reservoir. We have rodents. Man, they show up in practically everything. Rats, mice. We got prairie dogs. Those are examples of rodents. Here's a dog. Dogs are very susceptible to Lyme disease right here. And then, of course, there's a human. All right? So here's how it works. This tick is feeding on that deer. All right? So what happens in the spring, the female tick leaves the deer. Actually, she drops off on the animal. And in early summer, she lays eggs, and these eggs hatch. And what happens? Larvae emerge from this egg, from these eggs. These larvae feed on mice. And here's where the big problem's at. This is the animal, the primary one, that actually has this organism, Borrelia burgdorferi, that causes Lyme disease. So what happens, at some point, these larvae leave the mice and they overwinter on dry leaves, right here, okay? And in the spring, they become nymphs. It's the nymphs that bite an animal or the nymphs that bite a human being. And this is where you see the symptoms, the bullseye or the bullseye rash or the target sign, all right? So dogs are very susceptible. Humans, of course, can get Lyme disease. All right. So we're doing good so far. All right. Ask questions when they come up. I need to hear you. I don't hear anybody asking questions, so I'm just going to move on.
I have a question.
Yeah, go for it.
Sorry, it's kind of like passed by already, but I was scared to
No
interrupt.
problem.
So
No, please, for the whole class, interrupt because I can't see you guys, right? So don't be afraid to just, hey, Bideshi, slow down. Let's go back to the slide. Okay, which disease?
oh, it was for the bubonic plague.
Yeah.
So the septicemic plague, is that associated for both the pneumonic and the bubonic
Yeah,
plague?
so here's what septicemia is. If you can break up the terms, it makes sense, right? Septic means really nasty. There's lots of toxin floating around. Emia refers to the blood. So what we have with, say, for example, with bacteremia, you have bacteria floating around in the bloodstream. That might not be a very bad condition compared to something like this. With septicemic, it doesn't matter if it's a plague or whatever. With septicemic plague in particular, you have the organism, Yersinia pestis, plus their toxins. It's toxins floating around in the bloodstream. So that's a very, very bad condition. All right?
And it's kind of like a complication of both.
Yeah, it is. You're right.
Okay,
So
thank you.
Okay, good. Are we good there? Other questions? All right. Yeah, don't be afraid to, because I can't see you. We're not in a classroom. Don't be afraid. If you have a question, say, hey, Bideshi, let's just go back and just slow down for a bit. Let's talk about this one some more. Okay, so I want to move on to the next disease. Yeah, go for it. That's a very good question. And it's going to solve part of the problem. Let me just say, we use rodents as the key model system, but in fact, you can find this pathogen in different animals too. In deer, for example, right? Because the ticks, they go back and they feed on the deer. On elk, you can find them in those. Even rabbits can actually have. But the primary reservoir would be rodents. Yeah, they're crazy critters. They carry a whole bunch of different pathogens, including one for viral diseases, which we haven't talked about yet. Are we good? All right. Now, moving on, there's, again, look at me for a bit. Remember what I want you to know, not to memorize everything on the slides. We have to put them there because to get a complete story. What I'm interested in, like I've been saying all along, names of organisms, disease they cause, signs and symptoms, something that's peculiar, like think about the target sign. Man, that's so specific for Lyme disease. We're going to see something similar maybe for fungal diseases called ringworm. Sometimes you see the same appearance, but here's the differential diagnosis. That thing doesn't spread so rapidly. So remember, we look at something called erythema migrans. It's expanding rash on the torso. You don't see that so much with fungal diseases. All right. Now, next on the list is relapsing fever. I want to say very little about this because we don't see a whole lot of cases anymore. But there's two different organisms that cause this disease. I'm going to focus on the primary one. It's called Borrelia hermsii. So remember, Borrelia burgdorferi causes Lyme disease. This one is relapsing fever. It's Borrelia hermsii. All right. It is a spirochete. In fact, this is what it looks like. This is a drawing. I'll show you some real pictures in a second. So again, check it out. The reservoir. Can't avoid them, right? Rodents, rats, and mice. And the vector to transmit this to humans, it is the tick. All right, so hold on, excuse me for a second. So we have the vector, it is tick. The reservoir, it's rodents. And other animals, but rodents are the primary ones. So here's what happens. Imagine here's an animal that's infected with this organism, Borrelia hermsii. And here's a tick again that feeds on that animal, then it bites a human being. Well, here's what's going to happen. Probably most people recover, so we don't know the stats for that. But when people do get relapsing fever, think about it. Relapsing, you see spikes and peaks and troughs and peaks, and that's what it means. It reoccurs over and over again. So most people who are infected, they would develop illness in about 2 weeks or so. So 5 days to 15 days post-bite. This is after the tick bites. And then what are the symptoms that you see? Well, fever, that's very general, right? But think about something like strep throat, if ever you had strep throat. So I want you to make these correlations. You get a fever, but you don't see relapses. You see fever, and then the disease subsides. Then you enter convalescence, right? But individuals who have relapsing fever, yes, they're going to have fever, of course. They're going to have chills, headaches, nausea, but then it's going to basically occur in cycles. So take a look at a typical individual who has a disease. Normal body temperature is about 37 degrees centigrade. So we're starting here. He has that tick. It bites a human being. Let's just say this is 2 weeks. What you would see is fever, chills, headaches, myalgia. So those are pretty much common, right? And then it subsides. And then what happens again, they pop out in the blood. You see the same thing over and over again. Now, this can cycle about 13 times over several days to months. But again, of course, people might end up in the hospital, especially living in the Western world, where you do have access to medical care. In some areas, you don't, by the way. That's just a reality. It's a sad reality. And then what happens, even without treatment, sometimes people can recover here on their own. But keep in mind, this is a spirochete. Spirochetes are pretty nasty bacteria. Remember what they can do. Think about Lyme disease. They can actually hang out in you for a long time. When we look at syphilis, also caused by a spirochete treponema pallidum, that can last for a lifetime once you get infected. So again, the problem is this. These organisms can hang out in tissues for a while, all right? So why do you see these fevers and chills? Here's the reason why, because the organisms, they kind of pop out from where they're at and you find them in the bloodstream. Now check this out, right? Basically, we're looking at one of these spikes back here, these fever spikes. We're looking at these areas. If you're to collect a blood sample on that patient and you do a smear, this is what you would see. This is a pretty nice slide, by the way. You can see red blood cells in the background. Here's a white blood cell. That's a neutrophil. That's what it looks like. Although those granules kind of be following me. But take a look at this. Of course, of interest is this little spirochete here. Let me just say, this is Borrelia hermsii, right? But Borrelia burgdorferi just looks just like this. You can see these, and during this fever spike, I can see the bacteria in the bloodstream. All right. Now, there's another one, the endemic form. Right? This is caused by Borrelia hermsii. That's the most critical one. We can also see the epidemic form. So you see very few cases in a huge spike. Very, very infrequent in the United States. But you go to places like Africa or China, maybe parts of South America and Southeast Asia, it's not unusual to find this other one. It's called Borrelia recurrentis. So I'm not too concerned about this one. Borrelia hermsii is the primary one. All right. Again, this is the spike that you see. It's when the bacteria is present in the bloodstream. All right. Now, the last segment of today's lecture is going to focus on rickettsial diseases. All right. So again, remember what we're looking at: diseases that are transmitted by arthropods. In other words, things like ticks, mites, fleas, and lice. So I'm going to focus on some really important diseases that can cause actually mass killings or literally mass deaths in our history, especially during World War II. All right. That's epidemic typhus. But let's just talk a little about rickettsia. We've actually seen rickettsia-like organisms before. If you remember Q fever, way back when with respiratory diseases, right? That's caused by Coxiella burnetii. That's a rickettsial organism. So rickettsia are very special in this sense. They are all obligate intracellular parasites. We saw that term before. All that means is this: they require a living cell to grow. If you don't have a living cell, just like viruses, these are not viruses, but this is something in common they have with viruses, they would not be able to grow. So they are all obligate intracellular parasites. They're very small. You can't see them with the light microscope that we have in the laboratory because we can't resolve them very well. It might look like little dots. So you really can't tell what they are. Generally, we use electron microscopy to see them. For example, right over here. This is rickettsia inside of a living cell. So remember this. All rickettsias are obligate intracellular parasites. They require a living cell to grow. Now, here's the thing. The ones that we're going to be talking about, guess what? They're transmitted by arthropods. Lice, fleas, ticks, and even mites in some cases. All right? Don't worry about treatment here. So what are the major species? I have them in the subsequent slides, but let me just list a few for you. And again, I'm going to just focus on 4 of them, really. Rocky Mountain spotted fever, RMSF as it's called, is caused by Rickettsia rickettsii. All right? It's transmitted by ticks. And check it out again. Rodents. Yeah, these vermins, as we call them. They actually harbor a lot of different pathogens. Ehrlichiosis and anaplasmosis, these are the last 2 ones I'm going to talk about. We have scrub typhus. We see primarily in Asia it's maybe South America, but primarily in Asia. This is caused by Rickettsia tsutsugamushi. They actually changed the name a while ago. It's called Orencia tsutsugamushi, but I'll show you that in subsequent slides. And then epidemic typhus, responsible for many deaths over the centuries. Rickettsia prowazekii, epidemic typhus. And then murine refers to rats or rodents. Murine typhus is called by Rickettsia typhi. And then something we saw already, I'm going to talk about here, Q fever, respiratory disease that we saw earlier, caused by Coxiella burnetii. That should sound familiar to you. Remember, this is a pathogen, or this is a normal bacteria that you find in animals, especially farm animals like sheep and goat and cattle. All right? Okay. Now here, let me alert you. There's going to be a lot of writings on the slide. But again, what I want you to know is this: names of organisms, disease they cause, and peculiar types of signs and symptoms. They always show rashes on the skin. So rickettsial diseases are kind of, basically, they're codified by this. People who have these diseases would have rashes, all right? So they would have very high fever, they'd be nauseous, they'd be vomiting, so that's emesis, that's another word for vomiting. They'd have headaches, muscle aches, so those are the general things. But high fever is kind of characteristic. High fever and rashes on the skin, kind of characteristics for this type of disease. All right, so let's talk about the first one, Rocky Mountain spotted fever caused by Rickettsia rickettsii. Right here, this is the organism that causes that. Let me say again, it's transmitted primarily by ticks, right over here. All right, ticks, generally speaking, ticks. Fleas and mites can also transmit this, but ticks is the primary vector. So what do I want you to know? When the tick bites, incubation period is like about maybe 2 to 3 weeks or so, depending on how much pathogens you get. But here's what's going to happen. Individuals actually have a pink spot on the skin. It should be pink. All right. So this is called a macule. This progresses to a pink-red papule. And then, of course, what happens, all these papules, they can fuse to form a maculopapular rash. That's what we're looking at here. All right. That's called so we're looking at signs and symptoms now. So this is called a maculopapular rash. Now, here's something very strange about it. Most of the time, I guess I can't say most, but many times what happens, the rash actually spreads inward from the extremities to the torso. All right? That's important. So this is called a centripetal spread. It begins at the extremities. This is like the hands and the legs, and then it moves towards the trunk of the body. So that's kind of weird, right? The reason I bring that up, because the other one, epidemic typhus, is reversed. All right, very strange indeed. So again, you can read up on this. This is not significant for this class. Let's look at the other form. Epidemic typhus is caused by Rickettsia, not rickettsii, but Rickettsia prowazekii. So Rickettsia prowazekii causes the epidemic form of typhus. Now, this is not transmitted by a tick. Check it out. It's transmitted by the lice. This is the hair lice. It's Pediculus humanus, but nonetheless, it's the lice. That's this creature back here. Now think about this. People who live in crowded areas or who are confined to crowded areas, if I have lice that's infected with this pathogen, then pretty soon a lot of people are infected, right? So this is what we see back here. This is a picture of a mass grave. This picture was taken in 1945. Well, you can already guess what that is. This is World War II, right? And what we see back here, it's a lot of people who were in the concentration camps. They died because of something called epidemic typhus. Many, many cases like this, not just one camp. Again, here's why. You have infected lice, not ticks, right? Remember this. What transmits Rickettsia prowazekii is not ticks. It's arthropod, yes, but it's not ticks. It is lice, hair lice. And again, this spreads quite easily from one individual to the next. So think about, this is a very good case study to actually have you think about how people's immune state can influence diseases. Think about this. I mean, nobody wants to be in a prison camp, a concentration camp during the Nazi era, right? First of all, they're malnourished. Their immune system isn't as it should be. And now you put a pressure on these individuals. So again, that pressure would be that lice that you see in a population like this that harbors this pathogen called Rickettsia prowazekii that causes epidemic typhus. And then it spreads like wildfire. So people, they have very high fevers, kind of characteristic for this disease, rashes on the skin. severe headaches. It's a very terrible condition to be in, all right? Also, just, if you know your history, Napoleon had to retreat from Russia. Guess why? Many of his troops actually died because of epidemic typhus. One bacterium, Rickettsia prowazekii. Now, there's another form of this disease that we see. It's called Brill-Zinsser disease. And let me just say, it's a relapse of relapse of this condition. It tends to be a lot milder than the original form of epidemic typhus. But years later on, what happens? Apparently, this organism can stay in the system for a while. It can remain dormant. And as people get older, the immune system actually depresses a little bit. Now we see this organism pops up again. So it causes this is a milder form. Brill-Zinsser disease is a milder form of this should be epidemic typhus. All right, this is what we're looking at. Ricketts I can't even write you get me. All right, epidemic typhus. Now the next form is endemic typhus. This is also called murine typhus. Every time you see that word murine, think about rodents. All right. So again, rodents are the reservoirs for this. What causes endemic typhus is Rickettsia typhi. Now, first of all, let's make sure that we understand this. Epidemic, very few cases, and a huge spike of cases that occur in a short period of time. Think about influenza. Every year there's an epidemic, and then that goes pandemic, right? Endemic simply means this: I have a low number of cases in a certain geographic area. It's not a spike. Let's just say there's 20 to 50 cases every year, and it doesn't change. That's something that's endemic. So in the United States, endemic typhus or murine typhus is prevalent. It's primarily in the southwestern part of the United States. But we don't see too many cases. Individuals can have high fever again, rashes on the skin. That's classic for this disease. And check this out. What transmits this organism is Rickettsia typhi. It's that flea again. That's the same flea that transmitted, guess what? Yersinia pestis. That's the one that caused literally bubonic plague or the pneumonic plague. All right. Now there are some other species I just want to mention back here. In fact, one only. So here's what we talked about. Just to do a little recap. Rickettsia rickettsii causes Rocky Mountain spotted fever. Rickettsia prowazekii causes epidemic typhus. Rickettsia typhi causes typhus or endemic typhus primarily in the southwestern part of the country. And the only other one I want to mention back here used to be called Rickettsia tsutsugumushi. Think about that name. It's kind of Japanese, right? So that name itself it seemed like a Japanese name. So this is primarily, you see this primarily in Asia and the Pacific, countries or islands in the Pacific. The name hasn't been changed. It's not Rickettsia anymore. So if you read literature on this, it's now called Orenchia tsutsugumushi. There are some others, but I'm not going to have you memorize those. All right. So the four I pointed out here: Rickettsia rickettsii, Rocky Mountain spotted fever, Rickettsia prowazekii, epidemic typhus, Rickettsia typhi causes typhus, and then of course we have Rickettsia tsutsugumushi or Orenchia tsutsugumushi causes scrub typhus, primarily in Asian and Pacific countries. All right. Now we're winding down. The last two I want to mention is the disease is ehrlichiosis and anaplasmosis. So here's the thing, they're also rash diseases, but what happens, these organisms can hang out for a while. And here's where they're hiding out. They're hiding out in white blood cells. So the first one is this one here. It's caused by, so this one is ehrlichiosis. It's also called monocytic ehrlichiosis. I mentioned that, we find out in a second. But the organism of the bacteria is Ehrlichia chafiensis. All right? This causes ehrlichiosis or monocytic ehrlichiosis. Here's what that means. This bacteria infects monocytes. You guys remember what monocytes are? If you've taken Bio 163, this is a blood smear. You can see red blood cells in the background. This is a monocyte. A monocyte it circulates in the bloodstream. It can leave the bloodstream to become macrophages. But in the bloodstream, here's a monocyte infected. See these little areas back here? These little inclusions? That's where the bacteria is at. All right. So again, this can hang out in bacterial cells for a while. Now the next one is called HGA. So HME is it's monocytic ehrlichiosis. Another one is human granulocytic anaplasmosis. So this is caused by anaplasma phagocytophilum. So granulocyte in this case would be a neutrophil. All right, don't worry about why we call them granulocytes. If you take a 163, again, that should ring a bell to you. We can classify white blood cells as agranulocytes and granulocytes, but I don't care about that. In this case, we're looking at anaplasma phagocytophilum. Let me just say the target cell would be this one here. It is a neutrophil, all right? So for ehrlichiosis, it's a monocyte. For the other one, anaplasma, it is a neutrophil. And again, you can see that pretty clearly here. Here's the nucleus of the cell, all right? And you see these little inclusions right over here. That's where the bacteria is at, all right? That's where it's at. Okay, so we see an increasing number of cases over the past 10 years. Before we had about 10, or actually about 2,000 documented cases east of the Rockies, where we see an increasing number of cases. Here's why people actually have been identifying these more. Sometimes people have chronic diseases and you really can't tell exactly what's going on. In other words, you can't isolate a pathogen. So that applies to Lyme disease and also syphilis, but in this case too, ehrlichiosis and anaplasmosis. So what you do, if you suspect this organism, you take the patient's blood and you look for antibodies against the parasite, against the bacterial agent, all right? Okay, and again, we see the same type of thing, fever, chills, muscle aches, nausea, etc. But it starts off as a rash disease here, all right? Serology simply means this. It's a blood test to look for antibodies against. All right, so we're done with chapter 13. There's one other chapter we have to talk about before we can schedule an exam. This is chapter 14. This is the last chapter on bacterial diseases. STDs, bacterial STDs like gonorrhea, syphilis, chlamydia, and then contact diseases. So we should be able to clear up most of that by next lecture, maybe the whole chapter, but I don't want to push too much information at a time. Because we have a lot of time for the we're done with half of the semester. We have enough time to cover everything. So I'm not too concerned about time. And then, once we get maybe midway through chapter 14, I say, hey guys, let's think about exam 3. We don't want to wait too long to have exam 3. All right? Okay, let me stop here. I can answer other questions that you might have. And don't feel bad if you want to leave. I already have you recorded. Your attendance is already recorded because you clocked in. All right? So on Wednesday, we're going to cover chapter 14. I'll decide what that quiz would be on. All right, so that'll be your 5th quiz. Thanks for asking, but it's not going to be on Wednesday. It's going to be on Monday. Are we good? All right, good. All right, guys, I'll see you guys on Wednesday, and have a good one.
Professor?
Yeah?
Professor?
Hello? Yeah, I'm here.
Were you gonna review the quiz?
This is quiz 4.
Oh, I mean, were you gonna review the quiz, the one that we just took on last week on Monday?
This is quiz 4, right?
I think so, yeah.
Yeah, we only have 4 quizzes. So I'm going to get out of this, and then I'm going to pull that quiz up. I think it's on there. I don't like to put things on the screen because people, please don't take photo shots of this. The quiz has changed, but still, the reason why I don't talk about exams, I can't pop them up on the screen. But I would cover the quiz because they're written anew every time.